Hello everyone, welcome to Mediverse Atlas. In today's video, we are going to talk about immunity. I want you guys to watch this video till the end because the basics for cell-mediated immunity and humoral immunity are in this video. So to understand these topics, the cell-mediated immunity and humoral immunity in detail, I want you guys to watch this video till the end. Now, let us start with the definition of immunity. So, immunity, it is the ability of body to resist and eliminate potentially harmful foreign substances such as microorganisms and toxins. That is, in simple words, it is the capacity of the body. So, it's the capacity of the body to resist pathogen. So, whenever a pathogen enters our body, when a microorganism enters our body, our immune system gets activated and it eliminates the pathogen. So this is the basic funda of this immunity. Now we look at the types of immunity. We all know that there are two types of immunity. One is innate immunity and other is acquired. So innate immunity, it is natural or non-specific. It is present from birth and it acts immediately against the pathogen and it has no memory. Whereas acquired immunity, it develops after exposure to specific antigen and it has memory. Again, acquired immunity has two types. One is the active immunity and other is the passive. Okay, now what is the main difference between this active immunity and passive immunity? So this active immunity, it involves the production of antibodies within the body itself. That is, antibodies are produced by body. Suppose, take an example, you have taken a COVID-19 vaccination. After you have taken COVID-19 vaccination, your body identifies that there is something foreign particle entered and it generates specific antibodies and it creates some memory. So this active immunity involves production of antibodies within the body. Whereas in the case of passive immunity, antibodies are received from another source. That is, take an example of a newborn child. Newborn child receives their antibodies from mother's milk, that is from colostrum, right? From colostrum they receive antibodies directly. Or else when we have a patient of a snake bite, antibodies are given directly to him. That is, anti-venom serum is directly given to him. This is also one of the famous example of passive immunity. I hope you understood the basic difference between active as well as passive immunity. Now let us understand the mechanism of innate immunity. So let us look at the types of innate immunity and their mechanisms. So first we have anatomical barriers. What are anatomical barriers? There are two barriers, skin and mucous membrane. Next, skin, it prevents entry of microbes and its acidic environment retards the growth of microbes. Okay, mucous membrane, mucus entraps foreign microorganisms and competes with microbes for attachment. See, this innate immunity, the mechanism of innate immunity is not much asked in our exams. So we'll just have a rough idea on it. We'll not be going into the details of this innate immunity. But mechanism of the acquired immunity is very much important. Okay, so for mechanism of innate immunity, we'll just go superficially. Okay, and we have physiological barriers are temperature, low pH and chemical mediators. How does the temperature regulate or how does the temperature provide the innate immunity? So normal body temperature inhibits the growth of pathogen and fever also inhibits the growth of pathogen. And low pH, obviously our HCl has low pH, so that is, it is acidic in nature. So the most ingested microbes are killed because of this acidity or the low pH. Next, chemical mediators. We have chemical mediators such as lysozymes, interferons, and there are other substances produced by leukocytes also. These also induce innate immunity. Next, phagocytic barriers. There are some specialized cells in our body or the WBC that are monocytes, neutrophils and other tissue macrophages. They also engulf the microorganism and cause phagocytosis. Okay, and the fourth one is the inflammatory barrier. This is somewhat important. See, it is these inflammatory barriers are more important in the point of MCQs. Okay, so what happens in this exactly this inflammatory barrier is when there is a tissue damage or an infection. Suppose think this as a tissue. Think this as a tissue or a blood vessels. So this is a tissue, this is a blood vessel. So when there is an injury, so if there is an injury here, there will be damage to tissue as well as to the blood vessel, right? When there is damage, there will be leak out of vascular fluid. Due to the leak out of vascular fluid, in the vascular fluid we have some chemotactic signals. What are these chemotactic signals? There are some chemicals. What are these chemicals again? We have some chemicals like serotonin, histamine and prostaglandins. So serotonin, histamine and prostaglandins, what do? They influx the phagocytic cells near the affected area, and this process is called diapedesis. Okay, I hope you are clear. When there is a tissue injury, there will be leak out of vascular fluid. Along with vascular fluid, serotonin, histamine and prostaglandins are released. They influx the phagocytic cells near the affected area. Now we'll look at the mechanism of acquired immunity, which is the most important one. Next, first, humoral immunity. In this video we'll just discuss the basics. In the next coming videos we'll go and discuss in detail. First, humoral immunity. It is mediated by B lymphocytes and produces antibodies that neutralize antigen, and it acts against extracellular pathogens. Steps we'll be discussing in the next video. Don't worry about the steps. Next, what are the types of B lymphocytes? We have two types of B lymphocytes, plasma cells and memory cells. Plasma cells, they generate the antibodies. Okay, so they synthesize the antibodies, whereas memory cells, they create a memory of the antigen. If there is another encounter of the same antigen, these memory cells get activated and kills the pathogen. Okay, next, mechanism of cell-mediated immunity. Now it is mediated by T lymphocytes. No antibodies are involved, and it acts against intracellular pathogens. See, cell-mediated immunity, it acts against intracellular pathogens, whereas humoral immunity, it acts against extracellular pathogen, and here antibodies are produced, but here no antibodies are involved, so antibodies are not produced. Okay, now let us look at the most important thing, the type of T cells. We have totally 4 types of T cells. The first one is the helper T cells. They are also known as inducer cell, or they are denoted as TH cells, or they are also known as CD4 cells. Now what is this CD4 plus? The CD4 plus is a marker. Now again, what is this marker? See, suppose this is a T lymphocyte. On this T lymphocytes we have a specialized protein present. Okay, and this protein is the receptor molecule, or the receptor protein molecule present on the T cells. So if the receptor molecule is of CD4 type, it will be helper cells. Now if the receptor molecule is of CD8 plus type, it is of the cytotoxic cells. Okay, so we have when we look at the types of T cells, we have helper T cells. Helper T cells has a receptor protein of CD4 on their surface, whereas this one, cytotoxic T cells, has the receptor protein of CD8 plus on their surface. Okay, I hope you got this point. Again we have types of helper T cells. T helper cells are also divided into T helper 1 cells and T helper 2 cells. Okay, now what does T helper 1 cells do? They are concerned with cellular immunity, that is, they are concerned with cell mediated immunity, and they secrete interleukin 2 and gamma interferons. Now what does this interleukin 2 do? It activates other T cells. Okay, and this gamma interferon, it stimulates phagocytic activity of TH cells. Okay, that is, this gamma interferon helps in killing of the pathogen with the help of TH cells. Okay, T helper cells has again 2 types, T helper 1 and T helper 2. 1 is concerned with cellular immunity, 2 is concerned with humoral immunity. 1 secretes interleukin 2 and gamma interferon, whereas 2 secretes interleukins 4, 5, 6 and 10. Okay, and these interleukins 4, 5, 6 and 10, they cause three things. They causes activation of B cells, that is they causes the activation of B cells, that is B lymphocytes. That is why they are involved in, they are concerned with humoral immunity. Okay, T helper 2 cells are concerned with humoral immunity, so they causes the activation of B cells, and they also cause the proliferation of plasma cells and production of antibodies by plasma cells. So these are the three main functions, or the three main things of these interleukins released by T helper 2 cells. I hope you are clear with this. Again, the second type is the cytotoxic T cells, that is they have a receptor protein of CD8 plus on their surface. Okay, we have earlierly discussed here. So these are the cytotoxic C cells, also they are known as killer cells. Okay, they are denoted as T8 or TC. Okay, the third one is the suppressor T cells, and the fourth one is the memory T cells. So till here we have studied about the mechanism of innate immunity, a brief intro about humoral immunity, and the types of T cells. I hope you are clear with this till now. If you have any doubts till now, please do comment in the comment section. And we'll be talking something about antigens and antibodies. Now, what is an antigen? Antigen is a high molecular weight protein or polypeptide which induces specific immune response in our body. So from this, what can we conclude? Antigens are responsible for the production of immune response in our body. Okay, so antigens are responsible for production of immune response in our body. Now, what is an epitope? See, these definitions are just for a basic idea to have a clarity on the concept. Okay, these are not much asked in exam, mainly they are asked in viva. So this epitope is a part of antigen which has ability to generate antigenic response. And antibody, what is an antibody? We have all studied in lower classes. It is a Y-shaped molecule, it has heavy chains, light chains. So antibody is a glycoprotein that is produced against the antigen, and antibody binds to the epitope. This is the main point here. Antibody, it binds to the epitope. See, we have certain pathogen, and this pathogen has antigens, and this antigen has something called as epitope. So epitope is the place where antibody comes and binds. See, this antibody, it binds to the epitope of the antigen which is present on pathogen. I hope you are clear. Okay, now we'll be talking something about antigen presenting cells. This is very important. Okay, now what are these APCs, the antigen presenting cells? Antigen presenting cells are specialized immune cells that process and present foreign antigens on their surface along with MHC, the major histocompatibility molecules, to T lymphocytes to initiate an immune response. I know you have not understood anything with this. We'll talk about it. See, we have T lymphocytes. Suppose think this as T lymphocytes, and we have something called as antigen presenting cells in our body. So this is an antigen. Think this as an antigen. Now, what is the problem here is T lymphocytes cannot recognize the antigen by themselves, so they require antigen presenting cells. What does these antigen presenting cells do? They take up the antigen, they process the antigen. Okay, so they process the antigen, and in the antigen presenting cells we have something called as major histocompatibility complex. Now, what is this major histocompatibility complex? This MHC, it is present on chromosome 6 of our humans. Okay, it is a protein basically. This MHC is a protein which is present in almost all the nucleated cells, so mainly in the antigen presenting cells. Okay, so antigen is engulfed by antigen presenting cells. Inside it process and fuses with MHC. Okay, so they fuse with major histocompatibility complex, and they are presented on the surface of antigen presenting cells. Now, as soon as they are presented on antigen presenting cells, T lymphocytes detect them and cause the immune response. I hope you are clear with this. Okay, so T lymphocytes cannot recognize the antigen by themselves. They require antigen presenting cells, and in antigen presenting cells we have some protein called as MHC. They fuse with the antigen particle and presented on the surface of antigen presenting cells so that T lymphocytes recognize them and cause specific immune response. I hope you have understood the concept. Next, see, these APCs help T cell recognize and respond to antigens. Next, what are the types of APCs? Totally we have three main types of antigen presenting cells: dendritic cells, macrophages, and B lymphocytes. Dendritic cells, they are present in skin. So the dendritic cells which are present in skin are called as Langerhans cells. Okay, and they are also present in lymphoid tissues. And what is the function of dendritic cells? They are most potent antigen presenting cells, and they activate the T cells. Next, macrophages, they are almost found in all the tissues, and they causes the phagocytosis and presents antigen to T cells. And B lymphocytes, they are present in circulating blood and other lymphoid organs, and they present antigen to T helper cells during humoral response. Okay, so B lymphocytes are involved only during humoral response. Please make a note of that. Okay, now next we'll talk something about MHC, major histocompatibility cells complex. Major histocompatibility complex, it is a cluster of genes that code for special proteins present on surface of all nucleated cells. Okay, we have talked here now. Next, these proteins help the immune system identify pathogen and present antigens to T lymphocytes. Here they presented to T lymphocytes, right? Next, and MHC genes are located on chromosome 6. Okay, and there are two types of MHCs: class 1 MHC and class 2 MHC. Okay, class 1 MHC presents antigen to cytotoxic T cells, that is, they present antigen to T8 cells. Okay, they present antigen to T8 cells. Remember, class 1 presents to T8. Remember 18, Virat Kohli's number. Okay, so class 1 presents to T8 cells, that is 18. Okay, and class 2, it presents to T4. Okay, that is 24. Surya's movie. So class 2 presents to 4. I mean the helper cells like 2-4. This is 18. I hope you got my point. So to make you understand, I have told these things. 18 is Virat Kohli's jersey number, 24 is Surya's movie. Remember this. So class 1 for T8 cells and class 2 for T4 cells. I hope you are clear with this concept, and if you have any doubts, please do comment in the comment section, and don't forget to follow our Instagram. I'll be providing the handle in the description. And thank you for watching this video. See you, bye.